Hello, friends. Welcome to session 8 of our book club series. Hello, this is the Friendly Pharmacy 5 channel. My name is Lindsay Dixon. I am a pharmacist and also the founder of this YouTube channel where we present evidence-based scientific information to help you know how to better advocate for your own health. And if you're a healthcare professional, to help you know how to advocate for the health of your patients with the latest evidence-based information. So this is the first series in our book club, and I was fortunate enough to be able to contribute to the book Gut Logic. So this is a book that was written by Graham McKenzie, who is also a pharmacist, and he is with us today. And he invited me to contribute a little bit to his book, and so it was a great experience doing that. But I also know that there's just so much interest and also information about the microbiome that's coming out almost on a daily basis. So I really wanted to create a space for those who are interested to go a little bit deeper on some of the topics that Graham outlines in the book, but take this approach from an evidence-based perspective, because on social media, you and I all know that's not always the case, right? So we are 8 sessions in, and it's just been such a joy to have so many incredible experts on the channel, including, of course, Graham McKenzie, who is the author of Gut Logic. So before we get started, I just wanted to share with you a fun fact about the microbiome. So did you know that different medications can affect the diversity of our microbiome? Conversely, the microbiome has an effect on the way the body breaks down medications and consequently has the potential to change the effectiveness of medications we take. Given the fact that dozens of drugs are co-metabolized by the flora found in contact with medications, we can consider the gut microbiome to be a metabolic organ within the body. And that is a direct quote from one of the chapters in Gut Logic by Graham McKenzie. And I just want to bring Graham up to the stage. Graham, you've been with us in all these sessions. You're here again on a nice Saturday afternoon. Thank you so much for being here. How are you doing today?
Doing very well, and I should point out that yourself and Dragon have contributed more than just small contributions to this book. They really help to tie the book together and help it make sense. And that combined with these sessions you're doing are CE quality information. So I really gotta thank both of you for the work you've done on this.
Oh, that's really nice. Thank you, Graham. And it's been a really fun project to do. I've actually never met you in person. People don't generally know that. I think the pandemic kind of brought us together. And your first book, actually, Healthy Logic, also is how I started, to be able to get to know you. So I want to let people know that if you do let us know in the comments, we would love you to engage in these sessions. Let us know in the comments where you're from, or if you have a question about the microbiome as we go throughout today's session. Every question or comment will give you an entry into today's draw to win a copy of Graham's books, so Gut Logic and Healthy Logic. And these are actual hard copies of the books. Graham sends them out himself. I think he signs them. I'm not quite sure about that. But if you do comment, no matter what platform you're joining us from today, because we're streaming from 4 different platforms, you will be entered into the draw to win a book, and we will announce the winner at the end of this session. So, Graham, going back to that comment that I made there in the "Did you know" slide, so our microbiome really does affect how we metabolize different medications, and you talk about this in the book. Could you just kind of elaborate a little bit on that? Because even just before the session started, you told me about some medications that I had no idea that the microbiome had anything to do with how we metabolize medication. So can you maybe outline that a little bit for our viewers?
Yeah, so there's a couple of ways. There's the effect that medications have on our microbiome. And then there's the microbiome's effect on the medications. So we've talked in previous sessions here on different medications and what they do to the microbiome, the most obvious one being antibiotics and uh, the extreme amount of time it can take to get back to normal from weeks to months after a course of antibiotics to get your microbiome back to normal. Dr. Demianos and Dr. Wishmeyer both talked about everything that you intake affects your microbiome. If you live with a pet, it affects your microbiome. But when you take a medication, like Dr. Wishmeyer had mentioned, the amount of C. difficile infections he sees, and It's really kind of correlates quite strongly with the explosion of PPI medications, which can lower your stomach acid quite successfully, but then there's no gatekeeper to keep the bad bugs out. So infections like that can take a little um, longer to go away. Statins on their own are just such a huge story with probiotic, both in how they affect your microbiota and how the microbiota affects them. So, when you take a statin, statins can actually be antimicrobial to certain species in the gut. And in turn, when they regulate the microbiota like that, there's a change in the metabolites that you get from the bacteria in your gut. And then that affects how the statin works in your body. So we know that there are certain bile acids that are made by certain probiotics that can actually mimic the statin and keep it from working when it connects with the cell. And some people just seem to have more than they should of those probiotics. And they're speaking now actually of having a test ahead of time to see if a statin would actually work for you based on that information. The other way around, when you look at the metabolism of drugs from probiotics, we look at oral contraceptives, and that's always been a recommendation from pharmacists when you get an antibiotic. Um, we see medications like I see about phenazopyridine, which we compound now in our lab in the store, and the degradation of that through hydrolysis in the gut can reduce the effectiveness of a medication like that. You can break down acetaminophen very rapidly based on your microbiotic level. And that slide that you have up there just really explains a lot of it, how your microbiota changes through age, if you're male or female. The medications that you're on have a huge effect. There's a study now that says 27% of non-antibiotic drugs used in humans can repress the growth of at least one gut bacterial species, which is really huge when you think of it, because most seniors are on at least 5, 6, 7 or more medications. And disease state, of course, as Dr. Wishmeyer outlined, a lot of disease states, particularly pertaining to the gut, can really have an effect on your makeup of your microbiota. So yeah, it's probably a topic that we could have done a whole book on, as far as those two things, what the microbiota does to the drug and vice versa. And it really opened my eyes when I was writing this book on how vast a topic this really is.
Yeah, it definitely is. Thank you for explaining that, because even the acetaminophen that you had mentioned earlier, I had no idea, right? And it just
Mhm.
goes to show how the information has evolved, fairly quickly, and it's even hard for healthcare professionals to keep up, right? Even when we're
Oh yeah.
trying to do so. And and I do know some healthcare professionals that have actually read the book, and I know you had said that you weren't sure if you could write this book for healthcare professionals. But there's been comments, from people saying, the book is really, really valuable, because it's hard to find, information about this from an evidence-based perspective, and in a way that it's been written in a way that people can really understand, right? So,
Yeah,
I think,
and that's the way the previous book was like too. Do I write it for the medical community, or do you and I write this book for on the gut microbiome for the public? Like, who's going to read it more? And then turned out, if you write it so that a 15-year-old can read it, then everybody will read it.
yeah, and that's been kind of the comments that we've been getting, right, from a lot of different people, right? Even one that you went to visit last week when you gave her the book, right? So,
Oh yeah, that was a nice little visit we had. I didn't get to the other one. They were in Washington State. It's kind of interesting you mentioned, do I sign the books? I sign all the books, and I have to apologize to the Washington State viewer that they got put into the envelope so quickly at the post office. I never got a chance to sign them. But if you want to send them back to me, I'll sign them for you.
That's great. Well, thank you so much, and I know that people really do appreciate, having, having won a book, those that have been participating, right? So,
Yeah.
that's been great. Well, Graham, thanks for joining us. I know, you're going to, be watching, the rest of the session with our expert, who is here with us, and, you'll be joining us during the Q&A after his presentation. So we will see you shortly, I guess.
Thanks,
Um,
Lindsay.
yeah, thanks, Graham. Without any further ado, I'm just honored to introduce you to our speaker today. Our speaker today is Dr. Peter Lin. Dr. Peter Lin is the Director of Primary Care Initiatives at the Canadian Heart Research Centre. He has a busy family practice in Toronto, Canada, and he is also a contributing author to the Canadian Diabetes Guidelines during 2013 and 2018 on the vascular protection section. Dr. Lin is also an associate editor for Elsevier WebPortal, PracticeUpdate Primary Care. Dr. Lin is also the health columnist for CBC, and he has lectured on a variety of topics, including the microbiome of both adults and infants. Dr. Lin, thank you so much for joining our Gut Logic series today. It's such an honor to have you here.
Thank you, Lindsay. It's a real pleasure to be with you and with Graham as well. I'm now amongst the real people that do microbiome stuff.
Well, we're doing our best to try to keep up, as I know you are as well. Dr. Lin, before you get into your presentation, I know you're going to talk to us about microplastics and GLP-1 and some other fascinating things about early life and the microbiome. But can you tell us a little bit about, you've been a practicing physician for quite a while. What led you into the field of medicine?
Well, it's interesting. So I started off in engineering, and then in those days, you had to do a couple of years in something in university, and then you can jump across to medicine. So I jumped across from engineering to medicine. My inspiration was actually Hawkeye from MASH, right? I thought he was very cool. I didn't realize there was a lot of hard work behind the becoming a doctor thing. And it was very interesting because as we learned these things, I remember getting the medical school or medical student diseases, right? You'd pick up every single disease that you read about. And then eventually I went into family practice, and it's been very fulfilling, the last 35 years, because you get to help people, but you get to help people understand things. So even if we couldn't fix things, we could still make people feel better. And I think that was very important for us to be able to do that. And in family practice, I would encourage anybody that has children or anything to push them into the family practice area, because we can do a lot of good, and not just the scientific stuff. We can do all of that, but we can also do a lot of the human part of it. And that's been very rewarding as well. As I've seen with you and pharmacies and all that kind of stuff, the same idea is that I know artificial intelligence is coming, but I don't want to deal with a computer. I want to deal with a person. And I think that's the beauty of being in primary care.
Yeah, that's wonderful. And I was researching you a little bit before our session, and I found an interview that you did, I think 9 years ago. I think it was for the University of Toronto. And
Right.
9 years ago, you were talking about preventing disease, which is something we don't hear a lot about. And I thought, wow, look, this was 9 years ago. You were talking about, I want to try to encourage people to take the necessary measures to prevent disease before it even happens. Do you think that we have made that transition in healthcare, or are we a little bit further in that direction? Um, could you maybe
I,
comment on that? Yeah.
I think we're still putting out fires very much. So we see the hospitals and things like that. So people still having heart attacks, strokes, cancer, those kinds of things. So we do need to do a bit more, especially seeing how everything is under pressure now. So that endpoint, if we could stop the flow, like the waterfall from coming, like people getting diseases or delaying diseases, that would really help out. So I think if we can spend some time on the prevention, which means primary care, pharmacists, nurse practitioners, patients, the more we learn about how we can run our body well so that it doesn't break down, then we don't need the mechanics as much, in the hospitals, and therefore we can take a little bit of pressure off of them. So I think that we can all do a little bit better. It's kind of like your car, right? You sort of take it in for maintenance. You kind of know not to abuse the car. And so maybe we need to learn that about our bodies as well. And I think microbiome is one of those areas where I remember in medical school, they just told us about the microbiome. There's this bunch of bacteria that hang out with you. And it made it sound like they were just there as sort of freeloaders almost, right? Not the ones that are working for us. So I think our perception of how our body works has been changing over the years. And perhaps that'll teach us how we can keep healthy by making sure that all these components are working well together.
I agree. I remember when I first heard that, your microbiome weighs about, I think, 5 pounds or something like that. And I thought, well, that's a lot of weight to be carrying around. I mean, I hope it's useful, right? But our knowledge has definitely progressed since then. So Dr. Lin, I'm not going to delay anymore. I know you have a presentation for us, and I want to just give you the platform. Again, as you listen to Dr. Lin today, if you do have any comments or questions, we will be doing a Q&A at the end of his presentation. So please don't hesitate to put in your comments or questions, or just let us know where you're watching from. We'd love to hear from you. And Dr. Lin, I'm going to give you the platform, and we really look forward to learning from you today.
Great. Thank you very much, Lindsay, for this opportunity. And it's always nice to hang out with you, because I've been watching your videos. You're so eloquent in speaking and also scientific, which is very good. So I hope you can see my slides. So Lindsay, I think you told us that the next time we meet, it'll be in a place like this where we can run it from a nice, beautiful beach. I hope you can see that. These are my disclosures, just to go over that I have worked with many companies before in education. I don't do any clinical trials, and I don't own any patents. So this is the beautiful coral reef. So our microbiota, our microbiome, is supposed to look like this: diversity, lots of different life, lots of alive. And as we know, the reefs are not doing well, so maybe our microbiome isn't doing as well. And if you look at the gut microbiome, like all the way back, it says all diseases begin in the gut. This was Hippocrates in around, somewhere in around 370 BC, saying these kinds of things, and that health is determined by the microbiota of our gut. So this is way back then. We didn't know anything about this, and yet they were talking about how important the microbiome is. And of course, we've got all these scientific things saying how important these bacteria are to us in terms of keeping us healthy. But let me just share something with you. So this is your DNA. So that's your blueprints of life. And from there, we make every part of our body, like we make all the proteins and everything else. So the way it works is that you take the blueprint and you make a temporary copy of it. And this is the messenger RNA. So this is a temporary copy of the blueprints. And then you go to the factory and you make a protein. So we have tons of these little segments that we can make these different proteins. And those proteins will become the structure of our cells. It'll become our hair, our skin, enzymes that we need to do chemical reactions, receptors to pass chemical messages and things like that. So everything is sort of built off of this particular system. And I wanted to show you how impressive this system is. So in one cell, to produce all the things that it has, it needs about 1,000 of these mRNAs, these temporary blueprints that have to go to the factories back and forth to make copies of the proteins that we need. Unfortunately, about 50% of them are destroyed every 30 minutes. So if you do the math, we have to make one of these messenger RNAs, these copies of the blueprints, every 3 seconds. So that's how fast your cells are moving. And every single one of your cells has to move at that speed in order to keep the cell functioning well. Okay, so therefore our body is doing an incredible job. And then what about the proteins? Like, how many proteins are we making? So this is research from Toronto. Basically, they looked at yeast cells, and they estimate there's 42 million proteins inside a simple yeast cell. And the yeast cell only has 6,000 genes, whereas the humans have somewhere between 20 and 25,000 genes. So way more genes, way more proteins that we need to make for our body. So therefore, it's going to be way more than 42 million. And so my point from all of this is that there's a huge amount of factory work that's happening within each cell just to keep us alive. And so therefore, we need to understand how that system works. And our own machinery is not quite enough. So in other words, our little assembly line that we have is not enough to do that kind of a vast amount of job. And so this is where the microbiome becomes very interesting. So if you look at the number of cells that we have in our body, so the total number of cells is about 10 trillion inside our body. And mostly it's red blood cells that are floating around. The total microbiome that we have is 100 trillion. So it's about 10 times more. So for every one cell that we have, there's 10 more of the microbiome types of people living with us, okay? And the reason why that's important is, remember I said our genes, we have about 20,000 of those blueprint genes that we can make different proteins and enzymes. That's not enough for all the things that we need. And so therefore, if you look at the microbiome genes, how many do they carry collectively, all those little bacteria? They have about 3.3 million genes. And if you think about each gene making a protein, which can help us in some way, an enzyme or some structure or some nutrient, you could see that for every one gene that we have, they give us about 150 genes. So which means that we borrow their processing power. So we're not able to process everything. So therefore we're outsourcing, right? We're outsourcing our work to the bacteria, and the bacteria are helping to do many things for us. They're helping to protect us. They're helping to give us nutrients. They're helping to break down chemicals that we're getting exposed to. So the chemicals and things don't actually reach us because they're in the way. They're between us and the real world. And so therefore, the microbiome isn't just a freeloader hitchhiking with us. It's actually our factory, our main factory that's producing all the things that we need. So that's why we have to pay it special attention. Now, there's going to be different microbiome in your mouth and in your nose and on your skin, but most of it is going to be in your intestinal tract, because that's the part that you face the outside world the most. So wherever you face the outside world, all these bacteria are helping you. So your nose is facing the outside world because we're breathing in stuff. Your mouth is facing the outside world. And the whole intestinal tract is actually facing the outside world, from your mouth down to the other end, which I won't mention. Basically, all of that is outside your body. And so that's why we have so much of the bacteria sitting there, because they're acting as a buffer to take care of all the chemicals and everything else that may be coming in. So that's why, instead of looking at it as just bacteria that we need to, sort of carry with us, these are the folks that are actually keeping us healthy and protecting us. Now, if you go back to breast formula or breast milk, I just did a whole presentation on breastfeeding and why it's so important. So we always say breast is best, but we never explain why is it the best. Okay, so let's go through a few things to explain why it might be really good and how it ties in with the microbiome. Let me just show you this breakdown of what is in breast milk. Okay, so most of it is sugar, is lactose. Okay, so that's one type of sugar, and that's main energy source. And we have some fats and lipids and cholesterol that's carried across in there. And then we have this 8%. So this is the third most common thing. This is 55%. This is 28%. This is 8% of HMOs. Now, what is an HMO and why is it sitting there? I understand proteins and stuff like that, but what is an HMO? So it turns out an HMO is human milk oligosaccharides. Saccharides just means sugar. Oligo means 3 or more sugars hooked together. So these are human-made sugar molecules that are hooked together. There's over 200 types of them. And the weird thing is that we can't digest it. So as humans, we can't digest it, which means the baby cannot digest this thing. And therefore, there's no nutritional benefit for the baby. So why would we have something in breast milk that has no benefit at all for the baby? That doesn't make any sense. So then people started to look at what these things are really doing. So it turns out these HMOs, these oligosaccharides, are prebiotics. In other words, they are a fuel source for the good bacteria. So prebiotics are things that we feed the bacteria to make it grow. So in other words, probiotics is where we give you the bacteria. Prebiotics is we're giving you the stuff that's so that they can survive and grow better. So therefore, the good bacteria are encouraged to grow by this HMO. The bad bacteria, this HMO will kill them off. So in one sort of particle, you can see that it's fuel for the good guys, poison for the bad guys. And that's how you're tipping the balance in favor of growing a good microbiome. So in other words, the microbiome is so important that we put in food into the breast milk so that the baby can then generate a good microbiome. Now, because you have a good microbiome, then you're going to give good protection. And these are your intestinal cells. So these are the lining of your cells, let's say, from your intestine. And so therefore, they will provide good protection here. And so therefore, you'll have very good local effects. You'll have less infection, less diarrhea, those kinds of things, and you'll have better absorption of nutrients. At the same time, these things will pass chemical messages to your immune cells, so your army cells. So your army cells become trained as to what to attack and what not to attack, who's friendly and who's not friendly. And that's all because of these good bacteria that are hanging out here. So these are your army cells will then have systemic effects because they wander around in the rest of your body as well. So these army guys will not only help you at the gut wall levels, but as they move somewhere else, they will also know what to attack, what not to attack. So you could see that there might be benefits of that as well. The other thing that's found in breast milk is this MFGM, which is a fancy name for milk fat globule membrane. And I was wondering, this is a mouthful to say. And so basically it looks something like this. It looks like the Death Star, you know, from Star Wars, and it's got all these things sticking out of it. And I was wondering, how do you create something like that? So it's very simple. Here's the mammillary glands. This is the glands that produce milk in either humans or in animals. And if we were to zoom in on this section, so the milk is produced here, and then that will go out to the baby. And so if you look very closely up here, here's the apparatus in the cell to make this little ball. So let me just zoom in on this one in particular. So basically what happens is that this is the factory that will make this little ball of fat. So there's a ball of fat. And as you can see here, the ball of fat has a membrane around it, and it grows, it puts more fat into the middle of it. And then as it becomes mature, it now goes out through the cell membrane. So you see this little red line? So that red line gets wrapped around it as well. So now you get the cell membrane wrapped around it as well. And it turns out on that cell membrane, you have a lot of proteins and molecules that stick out in that membrane. So as it's pushing through like saran wrap, the saran wrap gets across it, and then you pick up all these little blue spikes at the same time. And all those blue spikes are components that the baby will need to build the brain, to build skin, and all those other things. So in other words, it's a complete package. And I often talk about it, it's like IKEA, you know, when you get the little package of screws and bolts, everything's in the right proportion. And that's exactly the same thing here, is that this is your package for your IKEA assembly. So this is what it looks like. Some of these spikes are called gangliosides, and these are useful to produce neuron cells, so your brain cells. And in the baby, the brain cells and the neuron cells are the ones that are the fastest growing, because they have to grow long, right? They have to stretch out. So it has to make a lot of these long membranes for itself. So therefore you need these things. Phospholipids are found in the membranes of all your cells. So as your cells are growing, you need phospholipids. So therefore it will help with growing more cells. Sphingolipids are also needed in brain development as well. So in other words, you've now got all the components sitting here, and it's being passed on to the baby. It has other components here, like other proteins, for example, mucin 1, okay? So mucin 1, it blocks the E. coli from binding. So E. coli, you might have heard of, you know, like the food contamination, and you get the bad version of E. coli, and then it makes people have diarrhea. So mucin 1 stops the bad E. coli from binding onto your intestine. So therefore it protects the baby against those kinds of infection. This one, lactohydrin, basically blocks rotavirus. So rotavirus is another infection that can cause diarrhea in kids. And the reason why it's so important not to have diarrhea in kids, because the kids will lose a lot of water very quickly, and they get dehydrated and they would die. So therefore, evolution has said, let us put in things that will help the baby avoid those early on infections so that they don't get dehydrated and die. So that's why that's all part of this package deal in the IKEA toolbox that we're sending across in the breast milk as well. On top of that, that MFGM has lots of other components. Here are some other components that it has. And these components may also be helpful in giving you the good bacteria. So the bifidobacterium is one of the good guys. And so what you see is that if you have the MFGM in your breast milk, basically the good guys increase. And then what about the bad guys? So the bad guy, E. coli, we talked about that, and Shigella, okay, those are bad guys, go down. So you can see that the bad people go down. These are the things that cause diarrhea. So this one molecule, as we're transferring it across, will be helpful in giving you protection in terms of the gut wall by changing the microbiome. So could we actually help to defend it? And that's really what it's doing. It's defending the child against infections. Now, on top of that, you can find certain bacteria that are helpful. So for example, this one, I don't know why they have so many Ks in there, but anyway, so this Akkermansia is a bacteria, and it's a very interesting one because it has some interesting benefits. So this guy is a gram-negative anaerobe, which means that it doesn't like to live in oxygen. So it likes to live in places with no oxygen. It's about 3% of your gut microbiome. It degrades bad mucin, so it breaks down the protective layer that's not good. And at the same time, the benefits is that it repairs that intestinal layer. So it breaks down the bad stuff, gives you back a nice layer. So therefore you can protect against germs coming across. It produces short chain fatty acids that help to become anti-inflammatory. So it reduces inflammation in the bowel area. So therefore we don't have wars down there. And then it modulates insulin resistance. And you might have heard about insulin resistance. So if the insulin is not working well, then your sugars are not going to be controlled, then you're more likely to have diabetes. And that's why when they look at studies in animals and stuff like that, you notice that there's improvement in metabolic syndrome, so insulin resistance things, there's less obesity, the insulin is more active, it's more sensitive, so therefore your body is listening to the insulin. The lipid metabolisms are better, and the liver metabolism, there's less inflammation. So in other words, all in good direction. And you'll notice that these conditions are things that we usually talk about in adults, right? So obesity, insulin resistance, diabetes, inflammation in the liver. So could it be that this particular bacteria is good for that? But could it be that it's good from the very beginning? So even at the childhood level, if we have that good bacteria, then we don't lead to those bad diseases. So this is one of the concepts. If we take care of babies very well in childhood, then can we avoid some of the diseases that we're talking about in the future? And so therefore the microbiome, getting it right from the beginning, it's a hint, right? Even in breast milk, they're putting in things that is there to feed the bacteria, and they're passing on things that kind of tip the balance against the bad bacteria and more towards the good bacteria, and protect against diarrhea and those kinds of things as well. How about the way that you're delivered? Okay, so they started, C-sections came along to save the baby's life or to save the mother's life, right? Because let's say baby can't get out, we need another way to get it out. So I think the C-section was a very good thing to help both mom and baby. But once they had enough C-section babies, they started to look at their health, later on in life. And they noticed they had more medical issues. They had more asthma, they had more diabetes, obesity. So they just had more medical problems if they were delivered by C-section versus the normal vaginal route. So that's when people started to think about, okay, so maybe there is something to the way that you're delivered. So therefore, if you were pulled out this way where you did not go down below, you might not pick up all the bacteria that you would normally pick up. So therefore, your microbiome may not start off with the starter pack that you're supposed to. And so therefore, you might not have all the good bacteria that you need. And so therefore, maybe this dysbiosis, so your microbiome isn't perfectly balanced, will then lead to more diseases like diabetes and obesity, those kinds of things. And so that's why they're trying to do studies. Should we take a swab of down below of mom and then put it onto the baby somehow, and then try and get them to have the microbiome? But exactly how much swabbing and where do you put it and stuff like that, it is a little bit complicated. But these are the things that people are starting to think about, because now we're collecting data saying that post C-section, the babies do tend to have more trouble. And now some of the baby formula people are saying that we can add in things to help with the microbiome. So people are looking at this, but certainly breastfeeding would be helpful, because then we would have all those other things that might help the good microbiome grow as well. And so the short-term complications, I guess, if you will, of C-section delivery are these kinds of things. So the short-term impact on the microbiome, and then the long-term impact, you can see a lot of these adult-type diseases that popping up: asthma, obesity, diabetes, that kind of thing showing up. And so could we tie it directly to the microbiome and a specific bacteria? The answer is no, but it seems like it's more common in C-section babies. So therefore, we have to take a little bit more care and study that. So again, if we can figure that out, we can prevent a whole bunch of diseases down the road. One of the thoughts was just give people the bacteria, like give it as a probiotic. So prebiotic is stuff that it helps to encourage the bacteria to grow. Probiotics would be things that basically we're giving you the bacteria. So the problem with that is that which ones that should we be giving you and how many? And how do we deliver it to the right places? Because as you can imagine, our small intestine is quite long. So somewhere between 18 and 20 feet long is the small intestine. And then your colon, the large intestine, is about 6 feet long. So that's quite a lot of real estate, right? So that's like the whole, edge of Canada, I guess, if you will, in terms of proportions. And so the idea is that where do we put them? How do we get them there? Or is it all over the place? Is it mainly at the top? Should we go from the bottom end, like with an enema, or should we take it by mouth? Or should we do both? So this is the part where we're not sure where we should put these folks. And so when people say, well, let's just put these three there, the concern that some people have is that if we just put those three, then those three may dominate. And so therefore it might muscle out the little guys that are trying to take up some real estate as well. So I think that's why this part needs a bit more thinking, so that we know what we're doing, so that we're not accidentally shifting the microbiome to less diversity, right? So if I give you, let's say I just give you one, but in huge amounts, then that one may grow and therefore muscle out other people. And therefore we've now shifted it so that that one becomes dominant, and then we lose diversity of the other bacteria that might be there processing off other things. So that's why I think this one needs some thinking, and we'll talk about AI at the end as perhaps the way to go. This is biodiversity. I'm not sure whether this is the way to get biodiversity. You know, people always say, you know, let your kids play in dirt and all that stuff. So I'm not sure if I want them to pick up all the dirt bacteria, but there are good bacteria that we would like them to pick up. We do want the bacteria that does all the processing and all those things that we talked about before, but I'm not sure kissing a pig might be the best way to go. So how can you help probiotics? There's a question mark in terms of the babies, what we should give them and how we should deliver them. Antibiotics, for sure, we want it reduced. You know, as Graham was saying, there's lots of antibiotics in our food. There's lots of antibiotics that we're taking for viral infections. So let's cut that down at least. Because the recovery time from there is a long time because we've wiped out a lot of bacteria. And then prebiotic might be a good strategy. That's the one like in the breast milk where it's encouraging the good bacteria to grow. So the prebiotic is like a buffet. I always tell people it's like a buffet for the bacteria. So that might be a good idea. Many times you're going to read about things like this, okay? So where they show you a graph like this and it says, you know, the microbiome is important for these toxins and this and this and this and this. And a lot of times it's so much stuff at the same time, it doesn't seem sensible almost. Like, how can the microbiome affect all of this? So let me just show you one example so that I can show you all the way down to the molecules as to what happens. And this one I'm going to talk about is Ozempic, okay? I know this has been a craze out there, you know, on TikTok and everything else. This is the Hollywood drug. And basically, I'm not sure if everybody knows, but I'll just go through the basics so that I know that I've done my job to sort of explain how this works. So Ozempic is a GLP-1 receptor agonist. So it stimulates the GLP-1 receptor. It's like a GLP-1 molecule. Okay, so what is a GLP-1? So let's go back here. So this is a class of drugs called incretins. So basically what I want you to know, this is your intestine. And in this purple part, this is the end of your small intestine, and then it connects up to your large intestine. Now, there is usually an intestine here. They just took it away to show you the back of it. So this is all connected. So this purple part right around here, there are L cells. And these L cells, basically, when they detect food, they will send out a message. And the message they send out is GLP-1. So they will send out this little molecule into the bloodstream, and it will go through the bloodstream to reach the rest of the body. So this is how it sends a message that food is here. If you think about it, your brain is very far from your intestine. It has no idea what's going on down there. And your pancreas has no idea what's going on down there. So we need a way to send signals to people. Now, if I was trying to send Lindsay or Graham a message, I would need to know their phone number, and I would type it in and send them food is here. The body is much smarter than that. Basically, this thing will just send out the message, and whoever needs that message will be able to grab it, okay? Because they'll have a receptor to grab it. And so basically, this is a text message that said food is here, and it will send it out, and whoever needs that piece of information will be able to grab it. So for example, the pancreas. I mean, the pancreas is very important, right? That's the one that controls sugar. It makes insulin and those kinds of things. So what would happen if the pancreas, let's look at what the pancreas does. So here's the pancreas. And in the pancreas, there are these cells that we have. We have the beta cells, which make insulin, okay? So therefore, if you're eating something, this guy is going to go up, okay? And then we also have the alpha cell, which makes glucagon. Now, glucagon is a hormone that will tell your liver to put out sugar. So in other words, insulin takes sugar out of your bloodstream. Glucagon is going to tell the liver to put sugar into your bloodstream. So therefore, when you're not eating, glucagon is the one that's keeping you alive because it's telling the liver to put sugar in. So now with those two systems, you can see I can take the blood out, the sugar out when you're eating, so when there's too much. And when you're not eating, like tonight when you're sleeping, glucagon is going to put sugar back in so your brain and everything else can keep working. So this is a perfect system so we can keep your sugar levels straight. But we need to know if food is coming or not. And so that's where the GLP-1 signal comes. So they send out a signal, food is coming. So now the beta cells know to increase insulin because food is coming. I'm going to need to take it out of the bloodstream and put it into the cells for storage. And at the same time, the glucagon can take a break because I don't need to put sugar in the blood because there's already sugar coming in from the food system. So you can see how the GLP-1 signaling is so important so that we get the right things happening at the pancreatic level. Now, what about your brain? Okay, so your brain might need that message as well. And why does it need the message? Is because if food is here, then it means there's no need to eat anymore. So therefore, it triggers satiety signal, which means I feel full. I don't need to keep eating anymore because I'm satisfied because there's food sitting down there. So this will control our eating as well. So therefore, the brain will need this message as well. And so how does this message happen? Here's the little receptor. Okay, so in other words, any tissue that needs that message will have a receptor. Along comes the GLP-1 molecule. The receptor will grab a hold of this molecule. And then when it grabs a hold of this molecule, it changes the tail part. And that's how the cell will know what to do. So if this is on the pancreas, it grabs the GLP-1, the tail changes shape. And then the pancreatic cell will know, okay, GLP-1 is here, which means food is here. That means I have to make insulin, or I have to stop making glucagon, or I have to stop eating. So that's basically how the signal is transmitted. And so therefore, this GLP-1 is going to have many effects on different organs, right? So the pancreas, I just showed you, the brain will be important, stomach, right, will be important, your liver is important, your heart is important if food is coming in or not. So therefore, they're all going to have receptors to grab the GLP-1 signal. And so, for example, if the pancreas, we just talked about it, insulin's going to go up, glucagon is going to go down. That's what it's going to do. The brain, we just talked about it. I've got food on board. I don't need to eat anymore. So therefore, I feel full. I don't eat anymore. Then I might lose some weight, okay, if I can keep giving you that signal. And that's where the injection of this thing would be helpful in losing weight, for example. The stomach emptying. So basically, if I know that there is food down at the bottom end of my intestine, then I'm not going to send food down there. So I'm going to slow everything down. So that's why you'll hear people, when they take those shots, that actually slows down their emptying of their stomach, and they actually feel very full that way as well. But you can imagine that if I'm on those shots and my stomach is slowed down, if I go for surgery, I may not get rid of the food as fast as usual. And so therefore we have to be careful, right? If people are taking those shots and going for surgery, they still might have food in their stomach. And so therefore we have to be extra careful with them. So this is all based on the signaling system that's there. In the liver, if there's food already down there, then basically I don't have to store as much fat and so on and so forth. So we're going to see some of these drugs being helpful to reduce the fat inside your liver. And then finally, these receptors are also found in the heart. So there may be benefits of the heart of this little molecule signaling. So that's why when the system is working well, all of these things are working. But let's say if the GLP-1 signal is low, now everything is messed up. So the body thinks that there's no food, so the brain starts to continue to eat. Your pancreas thinks that there's no food, so it doesn't make insulin. It keeps pumping sugar into the bloodstream, and so on and so forth. So you can see that if that signaling was gone, then everything would get messed up, and therefore we would see the effects of obesity, diabetes, those kinds of things coming. Now, how do we tie this to the microbiome? So this was a great paper in 2018 from Gut. And so what these folks said, here is your intestine. And remember the little L cell that I talked to you about? That's the guy that makes GLP-1, right? So there's the GLP-1, and that's the one that tells you to, lower sugar, you don't need to eat, all that kind of stuff, okay? Now, it turns out, who's telling the L cell that there's food around? Well, it turns out it's the microbiome. So the microbiome will produce these chemicals. These chemicals will work on some receptors that then tell the cell you should make the GLP-1. So not only does it give it signaling, it helps it to make the GLP-1 and then therefore give this. Now, what would happen if the bacteria are not there? So therefore, let's say we have a screwed up microbiome, then we don't have these little molecules that are sitting there in the right amounts, and therefore the signaling here is not in the right amount. And therefore you would tend to keep eating, thinking that there's no food sitting in the terminal part of your intestine. So you can see how the microbiome is helping us in terms of regulating whether we send out these signals or not. And I just show you this one example to show you how the microbiome that's sitting on this side of the gut wall can affect, what's happening inside your body. And in this case, it's affecting something that's happening very far away. Another half of this is that if these guys are not being produced, it affects the lymphocytes. So these signalings will also, remember, teach your army cells what to do. And therefore we affect the lymphocytes and their defense mode. So they might not be as good. And so therefore now we have people with, higher sugars, higher weight, maybe not so good at fighting infection. And we're now describing a patient with type 2 diabetes, for example. So therefore the microbiome may be, at least in the beginning, sort of initiating some of these problems that we see in diabetes. Now, I wish it was just so simple. We correct your bacteria and everything's good. Eventually, the system, it's kind of like having a something loose in your car. Initially, it's just the nut that's loose, but now everything is wearing out, and now we have more problems. So fixing that original nut doesn't fix everything, because now you've worn out other components of the system as well. Okay. What about the coronavirus? That did a lot of bad things to the microbiome. And I know that you guys had a whole session on that. I just want to show one slide here. These are patients with COVID-19, patients without COVID-19. And you can see the sort of the types of bacteria that are sitting there versus the types of bacteria that's sitting here in the non-COVID. So you can see many more of these ones, whereas these ones that are flagged here are very low in the non-COVID patients. So in other words, there's a population shift. And could that shift not go back to normal, in which case we might have long COVID bowel symptoms as well. And so that's what we're seeing. Some people have these bowel symptoms that continue on. And could that be because the microbiome has shifted? Also, if your microbiome has shifted, remember, it's processing a lot of things for us, and we need it for all that processing for internal systems. So then if they're not processing properly, then the internal systems are going to be worse. So we saw more diabetes, post-COVID. So one possibility is that it kills your pancreas cells. Another possibility, it's messing up that signaling with the GLP-1, right, that we just talked about, because it's messing up with the microbiome. So you could see that the microbiome would be very important to look at. Now, this one's interesting. So what about plastic? We were talking about this microplastic business. So it's showing up everywhere. You're seeing it on TV and stuff like that. And you've got all these little acronyms that nobody knows what it means. And so just to give you the background, everything is poly, meaning many. So it's polyethylene, which is PE, polypropylene, which is PP, polyethylene, and you can see that everything is poly, and then it's whatever the molecule is that it's making copies of it. Polyvinyl chloride, PVC pipes, we all know that one. And if you look at this one, this is what polyethylene is. This is ethylene, and poly just means many of these connected together. So plastic is basically many repetitious of the same unit that's here. This is propylene, okay? So this is polypropylene. So many of these things connected together, and these are the other ones that you can connect them together. And they have different properties. You can see them as styrofoam, styrofoam cups and things like that. Some are clear, some are not clear. So therefore, that's being used everywhere. And the problem is that we've been talking about it is in the nature, you see horrible pictures of birds and fish being tangled in the plastic thing. And we were always told that it's a long process, right? So we dump it in there, it breaks into small little bits, then the fish eat it, and then we eat the fish. So it makes it sound like it takes a long time for us to get the microplastics into us. Unfortunately, we are the first users of the plastic, right? So we're the first users, and then we throw it into the environment, and then the fish is like a second-hand user, and then we become third-hand user when we eat the fish. But we're the first-hand users of this plastic, so therefore it might make sense that we might have the plastics within our body as well. So this is a paper that was just published in March of this year, talking about microplastics in the carotid arteries of people that were going for surgery. So, in the arteries of your neck, sometimes cholesterol builds up. So this is normal looking. You could see cholesterol building up. So one way to prevent a stroke from happening is if it's really blocked, they go in there and they remove this. They actually take out that plaque and then put in new piping, I guess, if you will. So then you don't have a chance of popping and causing blood clots going up to your head. And so at this facility in Italy, they said, why don't we do that? But we'll send this to go look for plastic inside there. So they were wondering whether there's plastic inside there. So they used a big fancy electron microscope. And lo and behold, you could see these are chunks of plastic inside the plaque of these people, where they took it out. And this is the plaque outside of the macrophages. So we have macrophages. These are like army cells that have eaten up all the cholesterol. So you could see these little bubbles of cholesterol that it's eaten up, and it gets stuck in the plaque, okay? And so what happens is that you can see the plastic here outside these macrophages, your army cells. And then here, you could see it inside the macrophages. These are plastic that's inside, and you can see the oil bubbles that are inside the macrophages. So that's very concerning. So not only is it in there, it's also in the cells that are part of our army cells as well. Now, they looked at what type of plastic. So polyethylene, or PE, is the most common plastic in the world. And so therefore, about 60% of these people had it. And this study was, I think, 230 people or something like that. So 60% of them had this one in there. Polyvinyl chloride, or PVC, out of this 150 or whatever, about 30 of them had this one. So they had two plastics in them, okay? And 60% had one plastic in them. So is there inflammation in the plastic? Is it causing trouble? And so these are people without microplastic. These are with. And you could see without, there's very little inflammation, but with, there is much more inflammation going on. And wherever you have inflammation around plaques, we always worry that plaque is going to bust. And therefore, you're going to form a blood clot to seal it up. And then that blood clot flicks off or clogs up, and then you have a blockage. So that's why we worry about inflammation. Then they also looked at the collagen. That's what makes your skin, tight and, firm, I guess, if you will, with the collagen injection. So the more collagen you have in the artery, the tougher it is. It can withstand some pressure. So without microplastics, it's very good. Lots of collagen, and the red stuff is the collagen. On the other hand, patients with the microplastics have very little collagen, and it looks like this. So this pipe would be more likely to burst than this pipe. This pipe is tough. The other one is not. So therefore, that's of some concern as well. And then what about heart attack, stroke, and death after their surgery? In other words, they waited, 3 or 4 years to release this study. They said the people with plastic in them, did they do just as well after surgery, right? Because the whole idea of the surgery is to prevent strokes in the future. And what you could see is these are the people without plastic in them after the surgery. So their event rate of heart attack or stroke or death is very low. But you see the people with microplastic in them, their event rate is much higher. And so this is about 4.5 times higher risk than these people. So they all had the same surgery to take out that blockage, but the people with microplastic in them are 4.5 times more likely to have more problems in the 3 or 4 years after their surgery. So now this becomes something that we could use to identify people at high risk, because they probably have plastic in other places, and those other places might clog up and therefore cause heart attacks and strokes and things like that. And so what do you think happens with all this plastic that we go through? Well, the hot water bottle and stuff like that, the microwaving of your plastic containers that get very hot. And you know how when you take it out of the microwave, it's kind of soft? The bottles in your car on a summer day is very soft. And here's some holes in the styrofoam that were burnt by these very hot little balls of sesame seed balls, and microwave washing of plastic. Now, there are some that you can wash, but a lot of times you're now breaking down the plastic, and sometimes it becomes rough. And so that means some of the plastic has come off. Those ones will go into the water. And then the rough surface, if you put food in it, then you'll probably eat it. So anything with high temperature and plastic, you might leach out more of the plastic, or at least the sort of chemicals from the plastic. And what do you think it'll do to the microbiome? So the microbiome, basically, if we can find it here, then it's going to be either breathed in or you ate it in. So in the feces, they can see plastic inside there. And the problem with the plastic is that it allows a place for the bacteria to hang together, so that they can cause these biofilms, which is a layer of bacteria that hang together, and they can produce a lot of chemicals that may be harmful. And also the plastic may change, of course, the population of the microbiome, right? Because people that can hang onto plastic may not be the good bacteria. And so therefore we might get into some trouble that way. And then if we think about sort of looking at fruits and vegetables and stuff like that, I thought that would be safe, right? Broccoli and stuff like that. So they started looking at, do they pick it up in their roots or whatever? And so these, as you can see here, broccoli, carrot, lettuce, apple, pear, potato, and this is microparticles, pico amounts per gram. So you could see like 300,000, 100,000 particles. So in other words, even in our fresh food, there seems to be these microplastics because it's getting maybe in the water or in the air supply where it's being blown around or something like that as well. And then when they looked at infants and adult poop, I don't know who does these studies, but kudos to them for doing these lovely studies. This is how much they had of these two types of plastics, again, in nanograms. So these are very tiny amounts per kilogram. But then in babies, the numbers are much higher. So if you look at sort of per weight, they actually have much more in it. And they have more plastic bottles. They're exposed to much more plastic things. And so therefore, they might actually have a little bit more than the adults that we see. The good news, scientists have been looking at these things and they found that there are some plastic eating bacteria. So on these plastic bottles inside dump sites, they found bacteria that produces chemicals that can break down. So they have an enzyme, so PETase, which breaks down one type of plastic and it uses it up as a fuel. And so therefore it can break it down. And so hopefully we can break down the mounds of plastic. I don't know how to get the plastic out of the carotid artery or your bowels, but we do have at least some plastic eating bacteria that might be there. Who knows? Maybe in the future we'll have these kind of bacteria in our bowel as part of our microbiome because we'll need their services to break down the plastic bits as well. What about pollution? I'm just going to spend 2 minutes on this. When you have a very clean burn, like lots of oxygen, you just make water and carbon dioxide. So there should be no smoke. But when you don't have enough oxygen, you'll create these chemicals in between. And some of those chemicals are these particles that are there. So enough oxygen, you get this. Not enough oxygen, you create all that smoke. And this is the size proportion. So this is a red blood cell size, okay, in nanometers. This is a bacteria. This is our SARS friend, which is tiny, but it causes a lot of trouble. This is the large particles called particulate matter 10 microns. This one is usually filtered out by your nose hairs when you breathe it in. But this is the one that we worry about, the 2.5 size micron particles. It looks about the same size as bacteria. So what ends up happening is that your body ends up trying to kill it like a bacteria. So it pours chemicals on it to try and destroy it, but it doesn't die because it's just a chemical blob. So your body pours more chemicals on it to try and destroy it. And unfortunately, if you do that in your lung, what you do is you take the nice elastic tissue and you keep pouring those chemicals on it. It becomes holy like this. So this is no longer good lung. There's no stretchy bands anymore. It's just full of scars. And so this is what we call smoker's lung or COPD. So in other words, they breathe in those tiny particles. Your body tries to get rid of them by pouring chemicals on it, and it destroys the lung tissue there around it. What happens if that happened in your brain? Like if you breathe this stuff into your brain, they've now said that you either can breathe it directly in, which is, one way. The other way is that it goes through your blood. So it goes through your lungs, then those little particles goes through your blood, gets to your brain. Then your brain starts having those pouring the chemicals on it and this inflammation process. And that can cause damage to your brain cells and can cause buildup of, plaques and stuff like that, like amyloid. So then it looks kind of like an Alzheimer's brain as well. So these little particles, if they deposit anywhere, can cause inflammation. If it deposits in your plaques, it will cause inflammation because the thing will try to pour chemicals on it. And that might cause more strokes and heart attacks and things like that. So having the pollution side of things is no good either. So short-term effects of pollution are you get shorter breath and those kinds of things. But the long-term effects is all of these things, all your systems being affected. And what do you think about the microbiome? So you can either breathe it in or you can eat those little particles in. And so in your lung, if you breathe it in, those little particles will mess up your microbiome in your nose, in your throat, and in your lung as well. And then if you eat it in, it messes up your microbiome in your bowel and intestine. So therefore, now you've changed the bacteria growth in those areas. And so in the gut, it's going to break down that nice defense system that we had. So that layer of mucus and everything else is gone. And so now you let in chemicals and bad things, and then you start affecting every organ because your border guards are not working well. So this is why the pollution side of things also ties to it. So in a very strange way, everything ties back into the microbiome, right? So breastfeeding, everything still ties back into the microbiome. And before, when we're just juggling a couple of different bacteria types, right? We say, okay, lactobacillus is a good guy, this is the bad guy. Like there were single bacteria that we're talking about. Now we're talking about juggling many things, right? We're juggling now plastic, we're juggling pollution, we're juggling antibiotics, your foods that you're eating. The food themselves might have plastic in them as well. So how does that affect your microbiome? Now it becomes difficult. And this is where AI may be helpful. And because AI can keep track of a lot of different pieces at the same time. So recently they talked about, the AI being able to handle 200,000 tokens. And what does 200,000 tokens mean? it's about the same as 150,000 words or about 500 pages of text. So that means we could use AI to track all the different bacteria, what they're doing, track the particles from pollution, and then track your microplastic. So we track all of them, and then we put it together and say, what do we need to do with you to help you be better, okay, at this and balance you out? So there's a chance of that. I hope that they use AI for good direction, like in this case. I know this is a very funny picture. This is, and you thought your job sucks. So maybe the future is collecting poop, okay? So then we have a look at the microbiome that you have in your gut. So your gut has about a kilogram worth of it or so. And so therefore we can look at it and see what you're missing. And then in the future, maybe, you know, McCoy will wave something over so we don't even have to take an actual sample. And then we'll know what your microbiome is, we'll know what you're missing, and therefore we can maybe correct and what's too much, and then maybe we can correct that as well. And maybe that way we can keep that system working, because it is the largest functioning system in your body that's working well. And my final slide here is the it's a label, and it said the following. It says, remove child before washing. I'm not exactly sure what bad thing happened to the child, but obviously they had to remove child and make a label. So the concept is that the microbiome is essential to life. It's not just a hitchhiker's guide. They're not just hitching on for a ride. That we need them, and therefore we need to pay attention to them. So whether we're dealing with pollution, microplastic, antibiotics, the types of foods that we're eating, down to breastfeeding, for example, we just need to be aware. And to Graham's point about drugs and things like that affecting it, chemotherapy is one of the biggest areas. So many of the patients being treated for cancer get hit with a lot of chemotherapy, and that really messes up their microbiome. So all of them have either diarrhea, constipation, like their bowels are messed up. So imagine we just emphasize how important that barrier and that absorption of nutrients and processing is, and then we wipe it out with chemotherapy. So there are some places looking at how can we replace that microbiome nicely after chemotherapy and restart the system. So I think maybe it becomes part of our thinking as to how do we make sure that the microbiome is protected, because it's doing all of these functions in order to keep us healthy as well. Anyway, I will stop there, and let me just take off my screen sharing here. Okay.
Thank you so much, Dr. Lin. That was fascinating. I have been waiting 8 sessions for someone to explain the link between the microbiome and GLP-1, and you did it just excellent. So
I'm a simple man. I'm simple.
yeah, I've been waiting, and it was just it was a wonderful, wonderful moment there. So thank you. So many interesting insights there. Graham, any takeaways or questions for Dr. Lin before we open
Or
this
more
up?
importantly, corrections. Corrections.
Or corrections? And
Yeah,
we
no
also love
corrections,
your
no.
slides. Yeah, nice photos.
Yeah, Lindsay. Lindsay, I know you've probably gone through a lot of class time like I have as well over your life. That explanation about the MFGM and all the information on that membrane that goes to the baby. I have to say, of all the biology and chemistry and physiology classes I've taken, I've never really had that explained to me before. That was really interesting how that happens and how it makes breast milk even more valuable. I know that there's in the pipeline, there's now something they're always obviously trying to combine medications together now, 2 different types, and the GLP-1 with glucagon is one of them. I know they're trying to combine them with the SGLT2 inhibitors as well. Have you seen any information on the effectiveness of combining the GLP-1 with glucagon?
Yeah, so those studies are being done right now, and they're going to be combining a lot of things. So the way I want people to look at it is that we're trying to hack into the messaging system, right? So GLP-1 is one messaging system, glucagon is another messaging system, right? And then also GIP, all right? So people have heard of Mounjaro or tirzepatide. So that's the one that is now available. That's 2 messaging that it's doing. So the GLP-1 is from the distal part that says there is food down here. The GIP is saying that there's food at the top of the small intestine. So right at the top, there are J cells that when they detect food, they send out the GIP signaling that says there's food at the top. So now we have a way of saying there's food at the top and there's food at the bottom. So basically in patients where that signaling is down, If we replace the signaling, then that should correct the problem. So that's what medicine is heading towards, is figuring out the signaling system that the body is using. And if it's too much, we try to take it down. And if it's too little, we give you back more. So for example, you're going to hear about a lot of these interleukins, right? So there's antibodies against interleukin 6 and all these kinds of things to fight inflammation. So the word interleukin means between white cells. So these are the chemicals that white cells use to talk to each other. So let's say there's too much interleukin 6, the number 6 version. And so therefore we need to reduce that. So what we're going to be heading towards now in medicine is we're looking at what things are too much, and we take it down for you, and what things are too little, and then we're going to boost it up for you. So in this case, GLP-1 is low, therefore we're going to boost it up for you, right? GIP is low, then we're going to boost it up for you. So in some patients, the glucagon won't be, this glucagon signaling won't be right. So therefore, if we boost that up for you, it may actually be helpful. I know it's counterintuitive, but that's the thought process. And they have to prove it, of course. And that's why they have to still do all the studies. So it might look nice on a piece of paper saying this is this, but in the end, we'll do that. And what I'm hoping in the future is that we can check it. See, right now, we don't have an ability. We don't really check your GLP-1 levels and say, you're low. We just say you have diabetes, you're overweight, so probably this will work for you, right? So I think in the future, we're going to be able to say, we're going to check it. If it's low, I'm going to boost it up. Just like your thyroid, right? Your thyroid is low. That's why I'm giving you back thyroid. And then we fix all the problems that low thyroid has. Your thyroid is too high, so now I have to give you something to block the thyroid. So it's the same thing. It's just we're now getting into these messages that are between the bowel and the rest of the body, between your heart and this. And it's kind of exciting because it's no longer a mystery, right? It's not a black box anymore. We can see the messages going back and forth, and we can say we're going to help block this or enhance that. So keep your eyes open. There's going to be a lot of combinations. But I think if we look at it that way, then it won't seem like magical. See, right now, people just say, Ozempic, 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 it's magical, right? But once we understand how it works, you realize there's a lot of good science behind it. And then you realize that maybe I just need the GLP-1. Maybe I don't need the GIP, right? So then now we can figure out what you need as opposed to just this shotgun where everybody gets everything that's the latest on the market.
Yeah, interesting. And I think, and I'm not joking about this, I'm sitting here thinking the next big drug on the market is going to be the one that removes those microplastics from the plaque in your arteries. Like you could see that happening, right?
Right? Yeah. Because if there is these enzymes that we have, right? And I mean, I don't know how you get it there. Maybe you inject it in there. But the idea is that right now, we never calculated microplastics, right? We always talked about your blood pressure, your cholesterol, and whether your platelets are sticky, and therefore we give you something to make them less sticky. But we don't have anything to reduce the plastic, right? So, you're absolutely right. So whoever comes up with the enzyme that dissolves the plastic away will be very good because it's going to do it elsewhere. The other thing that I'm hoping they'll do with that study is to go back to those people and say, can we look at your liver and can we look at your intestine for the microplastic, because it's coming in from somewhere. And we need to figure out, like, what do they do? Do they chew on plastic, forks, or like why 60% of them have plastic and the other 40 don't, right? So from that little group, we should be able to get that. And what I'm hoping is some of our good Canadian institutions will start saying, can I ultrasound you and see the microplastic, right? So you can imagine if we do a very simple study, we do ultrasounds on people before, we send them for surgery because they need the surgery anyway to take away the plaque. And then we can take that plaque and we say, what did you see in the plaque when you looked with the microscope? Oh, we saw these little chunks of plastic. Then we can go back and say, on the ultrasound, could you see those little plastic spots that we saw in the real pieces? And if they can see it with this, then that means we have another way to predict who is going to do worse. So in that person, even though they've had surgery, I want them to quit smoking. I want their blood pressure to be perfect. I want their cholesterol to be perfect. I don't want them having diabetes, right? Because I can't fix the plastic, but what we're going to do is make everything else as tight as possible. So then the plastic risk I can't get rid of, but let's get rid of every other single risk for the moment. And then maybe you can do some tinkering in your backyard and come up with the plastic removal stuff.
Yeah. I mean, I could talk to Dr. Lin all night. So, Lindsay, you can go ahead with the questions you have.
Yeah, no, that's great. And Graham, I look forward to seeing that tinkering live on the channel,
Little
your
test tubes
production.
and everything else.
Yeah,
Yeah.
absolutely. I thought it was really interesting when you talked about these microplastics and the collagen, right, aspect, and I may have actually missed it, so I forgive me if I did, but do they know what the mechanism is? Like, is it an inflammation issue, that we see this lack of collagen in the people who do have the microplastics, or do we even know why that's
No,
happening?
no idea. So I'm hoping that they'll look at how many of the collagen producing, like the fibroblasts, like the actual cells
Mhm.
that make collagen, may have been injured, right? So in
Right.
other words, the microplastic makes them not produce, or the microplastic is getting in the way so that they can't make good collagen, because collagen is like a weave, right? So it has to be connected together properly.
Yeah.
So by having a chunk of plastic sitting there, maybe it's screwed up its production, and that's why it's so low. Like, I don't think they were expecting low collagen. They were expecting inflammation maybe, but not low collagen. But you can imagine those pipes are now at higher risk of rupturing even. And so, and repair, right? So you need collagen to repair. So now if, let's say, the blood pressure hits it and there's a bit of damage, they might not be able to repair. And therefore, maybe that's why those people end up getting into more trouble as well.
Yeah, no, it's really interesting, and I know collagen is a topic that as soon as you say the word right now, everyone's really interested, right?
Everybody's thinking, let's inject collagen. But
Yeah,
I mean, in this
exactly.
case, by figuring out the plastic story, we might be able to keep your natural collagen, right? Maybe you'll look younger. I don't know. Maybe your skin, because our skin is also exposed to quite a bit of air, and airborne small particles will have plastic in them as well. And so maybe if we protect ourselves, then maybe your skin will look nicer even.
Could be. If you are watching with us and you have any questions for Dr. Lin, this is the Q&A portion of our session. So you can just put your question into the comments section, and we will. We have Dr. Lin with us for a few more minutes, and we will ask him your question. So I just want to get to some of the questions here from some of our viewers. My mouse is just acting up. Just one moment here. Because we had some really great questions come in. This is an interesting question. I don't know if you know, Dr. Lin, do GMO foods help the gut? And I guess the contrary to that would be, what do they harm? Have you seen anything along those
Yeah,
lines?
no. And the problem is, to do that study, we would have to take people's bowel flora ahead
Hmm.
of time and then put them on the GMO food and then check their bowel flora afterwards again. And so that's why all of these studies are tough, right? Because when we look at with the bacterial flora, many of these bacteria do not grow in the air. So therefore, as soon as you take the sample, they die. So that's why we're looking at their genetic coding, right? So we look at their genes and we're probing for those genes to see if they are in existence or not. So that's why these studies are so difficult to do. And usually we have about 1,000 different types of bacteria. And the problem with trying to type 1,000 different types is difficult, right? So we usually can see the big sort of families, but it's harder to get down to the specific species. So that's why the research in this area is difficult, because we can't get the information so easily. Like, whereas if I want to check your sugar, I just take a blood sample, and I can get that, right? Whereas this one, I have to take a stool sample, we have to send it off, get the genetics tested. Now, what's interesting is NASA had done a twin study, where one twin astronaut went up to space, to the space station, for a year, and the other twin stayed down in the ground. Now, they did many things. They looked at blood pressures and everything else, but one of the things they did was they took samples of stools up there and down here, and there was a big difference in the microbiome of the two twins. And so now they're trying to figure out, do they get more diseases or not? So when he came back, his microbiome started to head back towards normal. And then now they're trying to figure out, over time, does one twin have more diseases than the other? So they continue to track that. So if ever you're interested, it's the twin study from NASA. That's a fun study to follow.
Yeah, that's fascinating. I have heard about that, and it's really interesting if people
Smart
want.
though. Smart. Imagine getting two twins as astronauts.
Yeah.
Go figure. It's like, not usual. Yeah. What do you want to be, an astronaut? You too? Yeah. Okay.
I could see them playing as kids. I
Just
wonder if it's because they ate differently though when they were up there. You got
Very
to
much
think
so.
all the different things
So
that can cause that.
that's right. So that's why they're trying to figure out, does space travel cause trouble? Does the type of food, because they're eating those little packaged foods, cause trouble? And how dramatic was it? So it's quite dramatic, right? Because the guy up there is not exposed to any bacteria. or any new bacteria. And then he's only eating these packaged foods. And then they're trying to figure out what solar radiation does as well to him. And also there's no bacteria in the food because they're all freeze-dried packaged. So therefore his microbiome would have shrunk. And then when he came back, the microbiome would have expanded again. And then the question is, having had the shrunken time, is he more likely to get arthritis and heart disease and those kinds of, so that's why they keep tracking these two guys forward. And then they can come up with, as our microbiome shrinks, right? Because I think some experts are saying we used to have 1,000 different types and now we're down to like 600. So depending on, like, let's say I only eat fast food, right? Then those bacteria will do well, but the other ones will drop off. So they're saying that we're now getting more narrow, and that means our processing ability is becoming smaller and smaller, which now means maybe more diseases are cropping up again. And then if I get an antibiotic or I get a chemotherapy, that now will really wipe it out. And so therefore, does that put me at higher risk of other diseases? These are like big questions. And it's one of those where you sort of say it makes sense, but then you got to prove it. So then we have to do stools on people and we have to, you know, do those studies. And I'm not going to do the studies. That's why we have very good dedicated scientists doing those things.
You can only do so much, right, Dr. Lin?
I'll explain the study.
Yeah. And that's also important. Teresa Croden has a question. I heard that when boiling water, the microplastics get stuck in the lime accumulating on the wall of the tea kettle. Is this a legit way to remove microplastics
from the water? And no, because it's only the stuff that's touching the wall. So if there is a reaction and the lime does pick up the microplastic, for example, the bulk of the center is not touching the wall. So if you're hoping that boiling would get rid of the water, microplastics, it probably won't. We probably need some sort of filter. So some sort of filter that we drink it through would be one thing. The other thing is that I don't want to freak people out, because I think our bowel and our intestine are quite good at blocking out things. So in other words, our bowel is not like a sieve, like it's not an open door where everything that we eat will come in. So I suspect that our bowel is rejecting a lot of this. So that's why in that study with the stool samples, you can measure plastic in the stool, but I don't think all of that plastic is going inside the body. So we would love to see if we can detect plastic in intestine. So I'm hoping that some surgeons that are taking out parts of intestines in people, like for a good reason, medical reason, then we can hopefully send those tissues off and look for plastic as well. And then for the people that are taking out chunks of liver for good medical reasons, that then we could send that off to look for plastic, because the intestine, if plastic gets into the intestine, the first place it'll go is the bloodstream will bring it to the liver. So the liver is where everything gets checked before it gets into general circulation. So if there's a pile of plastic here, I'm suspecting that either in the liver or in the intestine, we should see plastic getting stuck in there as well. The other place is lung. So I'm hoping that the lung surgeons, if they're taking out lung cancer or something like that, that maybe we can send some of those off to look for microplastic as well. So those would be very, very useful ways of figuring out where the plastic is coming into the body.
Thank you for answering that. That was a great question from one of our viewers, I think in South Africa. Nice comment here from Kim. Always like hearing Dr. Lin speak. CBC for sure keeps it real and easy to understand. Unfortunately, late to the show. So that's really nice to hear. We know Dr. Lin is very dear to us Canadians here. So if you have any more questions for Dr. Lin, we just have a few more minutes left. Just let us know. Graham, do you have any more questions for Dr. Lin?
As far as supplementing with collagen, is there any effect that would have on plaque formation with or without microplastic? Do you know? I know that when you're taking collagen, the body has to rip it apart and put it back together anyway. And as you get older, we're not that great at putting it back together in our
Right.
bodies. But
Yeah, and the problem is the location, right? In other words, it
Yep.
may, because we were trying to get it into joints, we were trying to get all sorts
Yep.
of stuff, and we can't target it, right? So all we're doing is we're giving you raw materials. So that would be like sending wood, and then you're hoping it ends up in your basement. And the problem is, I don't think we're missing the ingredients of collagen. So I think the problem that we're seeing there is that the plastic is getting in the way of how the collagen is being put together, as opposed to we're deficient in that material. So if you're deficient, then I give it back to you, then you'll be able to repair. But if you're not deficient, I think there's a problem with the plastic getting in the way. It would be really nice to look at the skin of these people, right? See, now all of a sudden I've got great ideas after the study's published, right? So if they looked at the skin of these people, are they making collagen in their skin properly? And if they are still making collagen in the skin properly, then that would be good. The interesting thing is that in the people that didn't have the microplastic, they had beautiful collagen. Like everything was perfect, right? So definitely the plastic is doing something to get in the way of the collagen assembling, as opposed to they're missing collagen or something like that. So I would suspect that the supplementation wouldn't fix the problem that they had in the plaques that we're talking about.
Okay.
All right. And I just had a quick question for Dr. Lin as well. So back to GLP-1, because, everyone wants to know. But I've heard about this Akkermansia, which you talked about. And I think there's no relation. I don't have any relationship with these people, but I know that there is a startup, I think, out of maybe San Francisco, and they're actually producing Akkermansia, which is actually very difficult to produce because it's anaerobic. So it has to be produced with no oxygen. And I guess the idea is that you could take a probiotic supplement, there's the way that they're marketing it, to maybe have some of these effects. What are your thoughts on this approach? It might be too early, but I just thought I'd pick your brain about it.
Yeah, so I'm always nervous where we find one and then we say, okay, let's just give you lots of that one, right? So the fear is that, remember, it's 3%. So let's say I go and give you 500 million, which isn't a lot, by the way, like in the grand scheme of things, not. But then we give it an advantage. So now let's say it grows to 5% of your microbiota. Is that a good thing to have 2% extra, right? So in other words, we want to replace back to normal. And the question is where on that 20, 2 feet of bowel or whatever, where do they need to go? Like, do they need to be distributed all over? So I just don't think that we know enough as to where to put them, right? Now, having said that, if somebody has a condition that is low in that one, right? So they have obesity, type 1 diabetes, those kinds of things, could we try it? I guess that would need a study of some sort to do. And I think the reason why I'm showing that that slide about how the MFGM inside the breast milk supports the growth of that is that there are other bacteria that the milk protein is doing that's supporting it, right? So in other words, it's not just that bacteria, it's just we've discovered that bacteria, like we found it and we kind of, some PhD student probably spent their whole life figuring it out and saying this bacteria is a really good guy. But there may be 100 other really good guys that we haven't colonized or cultured yet. So that's why I'm not sure about jumping in and saying, okay, this is the one that you need lots of and just take it. That one, I don't think we're smart enough to be able to do that right now. So in the future, if they show that, if you give it to obese people, type 1, they actually do better, then with that kind of studies, then maybe we can try. But right now, I don't want anybody going out there and finding something on the internet that just has the word there, and then they start popping these $35 bottles of capsules and stuff like that until we know for sure. So it's different to say that this guy is a good guy. It's very different to say I should give it to you. Okay, that's two completely different stories. So to say it's a good guy and that breast milk supports this good guy, I support breastfeeding for babies, right? I don't support going out and taking this particular bacteria right now.
Yeah, it's a really good point. Even since starting these sessions, I've noticed that on social media, I've been receiving more ads for these probiotic supplements, and one of them was a GLP-1, and it just kind of, or it's not GLP-1, but it was kind of, using that as a marketing tool. And I thought, oh no, here we go, right? And people are going to start being targeted in this way. And it's very important to make the distinction that you just made there. So,
and I think it's very easy for people to make we're like a GLP-1. And then to try and dispel that, oh my gosh, it takes so long, right? So in other words, people always believe the first fact that sounds really good to them, right? So this, like all these herbal GLP-1s, suddenly this is an Ozempic, but it's a herbal firm and it's only $25 or something like that. So those ones I get very nervous about for people following. So please, I like the way you guys are doing it. And Lindsay, you've done a good job. And Graham, you've done a good job. Sort of here's evidence. And then we base it off of that. Not just, I think this is good. And I did it for myself and it works. So therefore you should do it for. And please don't listen to people that are selling you something at that moment at the same time. It's very scary.
Yeah, we talked about this last session, actually, how, you take It doesn't matter if it's a probiotic or what it is, but Dragon has cited AE Probio, which actually gives you the evidence of where you have product A and you have medical condition B. You give the product, and statistically, you look at it. If it works, then it works, and if it doesn't, then it doesn't. So, to your point, when you find the one thing that you think is working, that's where you have to bring in that kind of a randomized placebo-controlled trial to see if there is more involved than just that one thing.
Exactly.
Absolutely. Well, I want to do our book giveaway, but before I do that, Dr. Lin, I want to thank you so much for giving of your time today to be here. I know you just got back from, I think, the Philippines. You were giving a talk, and you're just, you're very well known, well respected, and so thank you for coming onto the channel to share this evidence-based knowledge that you have. I also wanted to ask you, is there anything you would like to leave with our audience? We have an audience of healthcare professionals and the public, but people are really wanting to be, I think, more proactive about their health these days. And I know that that's something that you're quite passionate about as well. Is there anything a message maybe that you would like to leave our audience with today?
Yeah, I think just remember the microbiome is a big component of our life. So it's not just something that people talk about, right? It's actually doing a lot of work for us. And so therefore anything that can keep it healthy would be good. And generally speaking, things like, even stress and stuff like that, like we should try and lead a good life, right? So the idea is to have a long, happy life. And I think if we keep our microbiome happy, that'll be good. So in other words, think about the things that you're pouring into your system and how it might affect those little guys down at the bottom, because it might be helpful. And think about the things that you're breathing in. So think about the plastic bottles and all those other things that you're putting into your body. If there's a forest fire warning there, please don't go out and breathe all that stuff in, because once you get it in, we can't get it out. And please, double think about antibiotics. When it's necessary, antibiotics are great. But let's not use them, for viral infections and things like that. So let's at least be aware of it, and let's try and stay as healthy as we can. And in terms of chasing those promises, like eat this capsule, you'll be fine. If you're going to put it into your body, at least do some research from some credible places. And you guys have done a great job with this series, so evidence-based. And what that means is that we've looked at it properly. Like all the medications and the types of surgeries we do has been tested. And so whenever you put something into your body, just make sure it's been tested. When we go traveling, right, you always look up sites to see, is this a reputable hotel? Is it, a terrible area? So at least do some homework, and make sure that the places that you're looking up the information are respectable places, and not trying to sell you something. I think that's very important.
Yep, and it's tricky to do that, right? You really have to be
Now with AI and fake everything, like it could be Dr. Lin saying you should take this capsule in the future.
Yeah.
And so just remember, I will never say you should take this and buy it from this website. So that will never come out of my mouth at any point in my whole life, I think.
Yeah, those are some great points. So thank you so much for that. And I will get on to our book winner today. So our book winner today is from LinkedIn. So Cassandra Kent, you were late joining, but you joined in time, and I think that you might be a pharmacist, but you will be winning a copy of Gut Logic and Healthy Logic today by Graham Mackenzie. So if you can email me at friendlypharmacy5@gmail.com, Graham will send those books out to you quite quickly. So congratulations on winning a copy of these books. They're great resources, and they're all evidence-based, and they're not trying to sell you anything or market anything to you. So thank you, Graham, for making that possible during these sessions. I want to let you know what we have upcoming. So we do have another session actually coming up this Wednesday. And so if we can go to the slide of upcoming sessions, on Wednesday, we will be covering, these are all the sessions that we've covered. Can you believe that we've covered all of these topics? It's been such an experience. And so on Wednesday, we'll be discussing the gut-brain connection. Dr. Paul Wishmeyer, who was here a few weeks ago from Duke University Medical School, will be here again with us to discuss this. And I suggest you buckle up and show up on time, because it will be a loaded presentation. So looking forward to that. That'll be at 10 a.m. Pacific Standard Time this Wednesday. And if you can't be there for the live session, you can always catch the replay. So thank you so much, everyone, for joining us. Thank you to Dr. Lin. Thank you, Graham, as well, for being here again for another incredible session. I hope you both have a wonderful, wonderful weekend.
Great. Take care. Thanks for having me. Nice to see you, Graham.
Thank you, Dr. Lin.
Yeah.
Thanks.
Thank you all for joining.